So this is the Unit 4 lecture for microbiology, and this section is going to be focusing on the genome and the genetic material, some of the basic genetics in microbiology. When we look at the genetic material, the genome is a term used to refer to the entire genetic component. It's not specifying what form it's in, it's a very general term. The gene is going to be the sequence that codes for a specific polypeptide or RNA. So it's just a small portion of the genome. The genome is going to be made up of DNA, and in the DNA, your coding sequences are going to be made up of base pairs. These are specific nitrogenous bases. They are paired up as guanine and cytosine, which we represent as G and C, adenine and thymine in DNA, which is A and T, or adenine and uracil in RNA. The DNA does have specific ends to it. The 5' end is going to terminate with a phosphate group attached to the 5th carbon. The 3' end is going to terminate with a hydroxyl group bound to the 3rd carbon. And the strands are going to run anti-parallel to each other. So one is going to run in the 5' to 3' direction. The other is going to be running 5' to 3' in the opposite direction. And the ends of the DNA and RNA are important because this is going to have to do with how the enzymes are able to add on new base pairs. It's going to read it specifically in one direction. Much like when you read a book, you read the sentence left to right across the page and from top to bottom. The prokaryotic genome is going to be a little bit simpler than the eukaryotic genome. The chromosomes in the prokaryotic genome are organized into a molecule of DNA that's associated with proteins. Typically they're circular in bacteria, and there's usually only 1 or 2 of them. Because they don't have a nucleus, instead they will have a nucleoid region, which is just a localized area where the bacterial chromosome hangs out. Plasmids are separate small circular DNA molecules. These are going to replicate independently of the chromosomes. A couple common types of plasmids are the F plasmid, that is fertility. This has the instructions for bacterial conjugation. And then the R plasmids are resistance plasmids. These are going to code for genes that are resistant to 1 or more antimicrobial drugs or heavy metals. Both of these are very common. The resistance plasmids are becoming more and more common with our overuse of antibiotics. Bacteriocins are factors or genes for protein toxins. The bacteriocin is the actual toxin that's going to kill the same or similar species without the factor. This is a way that they can help to kill some of the competition. Virulence is going to be coded for in plasmids. Plasmids will have instructions for enzymes or toxins to be pathogenic. So there are some bacteria that are not pathogenic to us as humans unless they have a virulence plasmid. And that is what actually makes them cause disease in humans. The eukaryotic genome is a little bit more complicated. It's surrounded by a nuclear envelope, which is a double membrane that surrounds all of the genetic material. In eukaryotes, we have histones, which are globular proteins that are going to be used into packaging the DNA into chromosomes. DNA has a negative charge, the histones have a positive charge, and so the DNA will wrap around the histones. When it does, it forms these 10 nanometer beads of DNA wrapped around the histone called a nucleosome. Chromatin is going to be the form that you're going to have your loosely coiled DNA in most of the time in a eukaryotic cell. It's going to consist of nucleosomes clumped with proteins, and it will form 30 nanometer fibers. During cell division, you would form the chromosomes. The chromatid is the single leg of the chromosome. In eukaryotic cells, we do have some extranuclear DNA. And it's going to occur in the mitochondria, the chloroplasts, and in plasmids. So with DNA replication, this is going to make copies of the DNA, and the cell will carry enough DNA for synthesis. It carries enough energy for DNA synthesis in the high energy bonds of the molecule itself. So it doesn't need additional fuel. It's got its own energy source to carry this process out. I recommend watching these links on your own. I'm not able to put the links into the YouTube videos. That's been the problem with previous videos in the past, so I will just leave those out. So DNA replication is going to be semi-conservative. It's going to be composed of one original daughter strand, and then there's one new strand. So here at the top you can see you have your two original strands. When they split apart, you have one original strand and one new strand that's forming on there. With the initial process, it's going to begin at a sequence called the origin, and the enzyme's DNA helicase will unzip the DNA, and it's going to expose the nucleotides in a replication fork. Here you can see this is a replication fork where it's split apart. The DNA polymerase will bind the strand, and it's going to add nucleotides onto the 3' end. So you can see here that it's binding on. Your DNA polymerase is moving this direction, adding onto the 3' end. Here it's binding and adding onto the 3' end. So because of the anti-parallel nature, you're going to have a leading strand and a lagging strand. The leading strand is going to be synthesized continuously as a single long chain of nucleotides from the 5' to 3' direction. With the lagging strand, because it still does have to go in the 5' to 3' directions, instead you're going to have to synthesize short segments and then join them later. So you would have synthesized this strand here, then come back up and synthesize this little section, then move back up and synthesize this little section, and those are going to get joined together. So in synthesizing the leading strand, you'll have primase that will synthesize an RNA primer that's going to be complementary to the DNA template. This is going to provide that 3' hydroxyl that's required by the RNA polymerase. Then you'll have the triphosphate deoxyribonucleotides hydrogen bond to the complements of the parental strand. The DNA polymerase will covalently join the parent strand, and it will have energy from the triphosphate deoxyribonucleotides, and this will add on at a rate of about 500 to 1,000 nucleotides per second. So it actually reads quite quickly. The DNA polymerase is going to proofread, and it will find that naturally about 1 in 100,000 nucleotides is mismatched. The DNA polymerase will proofread it, and it will decrease the error to 1 in a billion base pairs. And finally, an enzyme will replace the RNA primer with DNA. To synthesize the lagging strand, you'll have a primase synthesize the RNA primers. Then the nucleotides will pair with the complements, and the DNA polymerase will join the nucleotides and proofread. These are going to be in the discontinuous segments. These segments are called the Okazaki fragments. They're about 1,000 nucleotides long, and each fragment is going to require a new primer. Another type of DNA polymerase is required to replace the RNA primer of the Okazaki fragments with DNA, and that will further proofread. So the DNA ligase is the enzyme that ends up joining the Okazaki fragments. So here again we have another animation that is helpful to be able to watch that I am not able to record in YouTube. So one of the things that helps to speed up the DNA synthesis is it can be bidirectional, so it's usually going to proceed in both directions from its origin. So when you create the starting point, it's going to be able to move in two different directions. This will involve two sets of enzymes and two replication forks. So in some cases you will find that there's been some methylation of the DNA. This is going to add a methyl group to one or two of the bases that are part of a specific nucleotide group. This is a way to help control genetic expression. It can help to turn genes on or off. It plays a role in initiating the DNA replication. It will help protect against viral infection. Viral DNA is not methylated, so when looking to try and destroy viral DNA, one of the things it will look for is to see if there's methylation, and it will leave it alone if there's methylation. It's also useful in helping to repair the DNA. When we look at the gene functions, the genotype is the set of genes that is in the genome of the organism. The phenotype is going to be the physical features of the traits of the organism. The genotype is the instructions that are underneath. The phenotype is what you actually will see. In order to express these genes, we have two parts that will happen, transcription and translation. With transcription, DNA is used as a template to make RNA, and then that RNA copy of the gene is what's going to be used for the synthesis of the polypeptides. And that synthesis of the polypeptides, or making the proteins, is translation. So with transcription, this is where we make the RNA. The mRNA is your messenger RNA. This is going to carry the genetic information from the chromosomes to the ribosomes. We also have ribosomal RNA, or rRNA. This will combine with ribosomal polypeptides to form ribosomes, so the rRNA itself actually folds up and creates part of the ribosomal structure. The tRNA, or transfer RNA, is going to be the one that delivers the amino acids to the ribosomes. It has a portion that is complementary called the anticodon that will be complementary and bond to the codon in a way of helping to make sure that you only bring the correct amino acid. So this process begins with initiation, where you have an RNA polymerase enzyme that's going to move along the DNA helix until it reaches a promoter. This allows the RNA polymerase to unzip and unwind the DNA in the promoter region. So here you can see it being unwound and unzipping. The promoter is a sequence that's near the beginning of the gene that helps to initiate transcription. Not all RNA polymerases bind to all promoters equally strong, and this will affect the amount and type of protein being transcribed. In bacteria, sigma factor is necessary. It's the portion of the RNA polymerase that is needed for recognition of the promoter. In eukaryotes, it's going to use transcription factors, which are proteins, to help bind the RNA polymerase to the primer. So they will have a similar type of function, but it's a sigma factor in bacteria, transcription factors in the eukaryotes. During the elongation phase, it's going to begin 10 nucleotides away from the promoter, and while transcribing those first 10 nucleotides, the RNA polymerase will release its sigma factor to allow it to adhere to the DNA tightly. Many RNA polymerases can concurrently transcribe the same gene. So RNA polymerase is different from DNA polymerase. When you polymerize something, you are making a polymer out of it, and you are adding to it. So RNA polymerase is used to make RNA, DNA polymerase is used to make DNA. RNA polymerases are going to unwind the DNA itself. It does not require a helicase. RNA polymerases are slower. They only move at about 50 nucleotides per second, and they're going to incorporate ribonucleotides instead of deoxyribonucleotides. Uracil will be incorporated instead of thymine. It also lacks the proofreading, so there's errors every 10,000 nucleotides. So during termination, the RNA polymerase and the transcribed RNA are released. So with self-termination, we've got a terminator sequence. The RNA polymerase transcribes the terminator sequence, which is going to be rich in G and C bases, followed by a region that's rich in A and U bases. It's going to slow down in the G and C region because it's going to make 3 hydrogen bonds between them. And this will also help to form a stem and loop structure that will actually put tension on the structure itself and then physically pull off in an adenine-rich area. With enzyme-dependent termination, it's going to depend on a termination protein that binds to a specific sequence near the end of the RNA transcript, and then the protein will move towards the 3' end and force the RNA polymerase and DNA apart. So there's a couple of animations here, again, that I recommend watching. So during translation, this is when you're going to actually translate the code to make the protein. The ribosomes will read the genetic information and synthesize the polypeptide. And here you've got a nice little illustration of this process happening that you may want to come back and refer to. So with our genetic code, it's nearly universal. There's a few exceptions. It is redundant, so you're going to have more than one codon associated with each amino acid. The codons are the triplets of mRNA nucleotides that are associated with the amino acids. We have 20 different amino acids. We have 4 different base pairs, so if you only use the 4 base pairs, that would only allow for 4 different amino acids. If we did them in pairs of 2, that would be 4 times 4, 16 possible combinations for 16 amino acids. Still not enough. So we have to do the codons in sets of 3, and that will allow 4 times 4 times 4, which is 64 possible combinations, which is enough to make sure each of the 20 amino acids is covered. But each of those 20 amino acids will usually have more than one codon that will code for it. AUG is the start codon. So the ribosome will go along and it will look for the first AUG to know where to start. This actually codes for methionine in eukaryotes and N-formal methionine in prokaryotes, which we just abbreviate as FMET. That will also occur in the mitochondria and the chloroplasts. In many cases, that methionine will actually be removed in the end when you process the protein, so that not every single protein in a cell is going to actually start with methionine. It can be modified in the end, but that's where we start reading the sequence. Then we have some stop sequences that are UAA, UAG, and UGA. These actually do not code for an amino acid at all, and that's how it knows to stop reading. The mRNA is going to carry an AUG to start, and then it will have sequential codons and at least one stop codon. The prokaryotic ribosomes are actually able to start translation before transcription is done. In a eukaryote, translation is going to be in the cytoplasm and transcription is in the nucleus. In a prokaryote, it's not sectioned off, so one enzyme can just hop on after the other one. It doesn't have to wait for it to move from one compartment to another. So there are some differences in the eukaryotes and prokaryotes here. The eukaryotic mRNA is going to contain non-coding regions, so it's considered a pre-mRNA. The introns are the non-coding regions. These will be removed to make a functional mRNA, and intron is just short for intervening sequences. Exons are the coding regions, which are expressed. Eukaryotic mRNA is going to carry instructions for only one polypeptide, where it may carry instructions for more than one in prokaryotes. And with the introns and exons, you can actually recombine those so one gene can actually code for multiple proteins. The eukaryotic mRNA is not translated until it's fully transcribed because it has to move out of the nucleus to reach the ribosomes. So the tRNA or transfer RNA is about 75 ribonucleotides, and it will form 3 main hairpin loops. It has an acceptor site where there will be a specific amino acid at the 3' end, and then you have an anticodon in the bottom of the loop. The anticodon is going to be the triplet at the bottom of the tRNA, and it's going to be complementary to the codon. So although there are 64 codons, some organisms have fewer tRNAs due to there being some wobble on the third nucleotide. If you look at a codon table, you will see that in a lot of cases there will be 4 different codons that will code for one amino acid, and the only difference is in the third nucleotide. This is what allows us to have this wobble in there. So it can bond to a nucleotide other than the normal one by changing the angle. And since the third nucleotide in a lot of cases will not change the amino acid, this lets the cell get away with having slightly fewer anticodons. So our ribosomes are also going to be different in prokaryotes and eukaryotes. They're going to be 70S in prokaryotes and 80S in eukaryotes. So remember that S is for Svedberg's, and it's measuring the sedimentation rate. Each is going to have 2 subunits. The smaller subunits will be shaped to accommodate 3 codons at one time. There's multiple sites on there. The A site is going to accommodate the tRNA delivering an amino acid. The P site is going to hold on to the tRNA and the growing polypeptide. The E site is going to be where the tRNA exits from. So when we look at the subunits of the ribosomes, the prokaryotes are going to have 50S and 30S subunits. The 50S subunit is composed of a 23S and 5S ribosomal RNA and 34 polypeptides. The 30S is composed of the 16S ribosomal RNA and 21 ribosomal polypeptides. In eukaryotes, you have the 80S ribosome that's going to have a 40S and 60S subunit. So you'll notice here that the size of the subunits does not add up to the total size because it's a sedimentation rate, not actually like a mass rate that gets added together. So the 80S has the 40S and 60S subunits, the large RNA and more polypeptides than you have in prokaryotes. So they're just bigger. Then we've got 2 animations of translation here that you can take a look at. So we have multiple stages of translation. During initiation, you're going to have the messenger RNA and 2 ribosomal subunits and tRNA form the initiation complex. The smaller subunit attaches to the mRNA with the start codon in the P site. Then the tRNA will attach to the start codon at the P site, and GTP is going to supply the binding energy. Then the larger ribosome attaches to form the complex. During the elongation phase, the tRNA with the matching is going to be matching the next codon will attach to the A site. The elongation factor protein will move and escort the tRNA with GTP, and the energy from the GTP will be used to stabilize the tRNA in the A site. The larger subunit's ribozyme is going to form a peptide bond between the terminal amino acid and the newly introduced amino acid. So this larger subunit will have an enzyme in there that's going to cause a peptide bond to form between the existing amino acid and the new one that's being brought in. The polypeptide attached to the tRNA on the A site is the one that's coming in. It will use GTP as its energy source, and then the ribosome will move down 1 codon and transfer the tRNA to the adjacent site. As it moves down, the ribosome will release the empty tRNA from the E site, and it can be recharged and pick up a new amino acid. The cycle will repeat itself, and it will also allow another ribosome to follow it as it elongates. So you can have multiple ribosomes reading the same mRNA to make a greater amount of the protein. So for termination, you have a release factor that's going to halt the elongation. It's going to recognize the stop codons, and it will modify the large subunit to activate the ribosome to sever the polypeptide from the final tRNA, and then the subunits will dissociate. So So in controlling transcription, about 75% of genes are expressed all of the time. Others are regulated so that the polypeptides are only synthesized when needed. So the operon is a way of regulating genes in prokaryotes. An operon consists of a promoter, an adjacent operator, and a series of genes which are going to code for specific enzymes or structures. The operator is going to be the regulator elements where a repressor protein can bond to it to stop transcription. So with inducible operons, they're not usually transcribed. They have to be activated by an inducer. A repressible operon is going to be transcribed continually until it gets deactivated by repressors. So here we've got an illustration that shows the lac operon. And the lac operon, or lactose operon, is considered an inducible operon. It's going to include a promoter, an operator, and 3 genes that are for the catabolism of lactose. This gets controlled by a regulator gene that's constantly transcribed and translated to make a repressor protein that's going to attach to the lac operator and prevent the RNA polymerase from moving beyond the promoter. When lactose is available, it will act as an inducer, and it's going to change the structure of the repressor and allow transcription to take place. When lactose is depleted, the repressor will become active again. So this is a way for the cell to be able to save energy. It will make the enzyme when lactose is present. It will stop making the enzyme when lactose is not present. And I'm strongly recommending watching these animations on the operons. They just tend to make a whole lot more sense when you watch a little video of it happening rather than just trying to read words or look at a diagram of it. So there's 2 different ones for the lac operon. So the tryptophan operon is another well-known one in bacteria. It's considered a repressible operon, and this is another way for the cell to save energy. This consists of a promoter, an operator, and 5 genes for enzymes in the synthesis of tryptophan. Normally the repressor is inactive, so when tryptophan is not present, the tryptophan operon is active. If tryptophan is available, it activates the repressor, binds the operator, and stops the RNA polymerase in transcription. So if tryptophan is available from the environment, the cell is not going to go through and make it. Again, the animations are usually very helpful in making sense of the operons. So mutations are changes in the nucleotide base sequence of a genome, particularly in a gene. We can have point mutations where just one or a few nucleotide base pairs are affected. So these can include substitutions where you will insert the wrong nucleotide, insertions where you will insert extra nucleotides, deletions where you have loss of nucleotides. Both insertions and deletions end up causing a frame shift mutation where the triplets subsequent to the mutation are displaced and it creates new sequences of codons. It essentially is going to change your frame of reference for reading, and you will end up reading entirely new codons instead of the intended ones. Some other types of mutations you can have are inversions, duplications, or transpositions, where larger chunks of DNA or the chromosome can be inserted in the wrong place, copied, or attached onto the wrong chromosome. Silent mutations are usually the best type of mutation you could have. Here the substitution does not actually change the amino acid sequence. It still codes for the same thing. It just does it using a slightly different sequence. A missense mutation is going to actually change the nucleotide, and it will result in a codon that will code for a different amino acid. So you will end up with a slightly different protein. This does have the possibility of making a better protein, but most of the time with a random mutation, it generally results in a non-functional protein. With a nonsense mutation, it's going to change the nucleotide to create a stop codon. This will usually make a non-functional protein because it's going to stop protein synthesis. Mutagens are the physical and chemical agents that are going to induce the mutations. Some mutations are spontaneous that result from errors in replication and repair. These are kind of like getting typos when you go to type a document on your computer. You can have recombination mutations. These are relatively long stretches of DNA that move along the chromosomes, and this will introduce frame shift mutations. So radiation is a type of mutagen. Ionizing radiation is going to include X-rays and gamma rays. These are going to bring enough energy in that will energize the electrons to escape and strike other atoms and produce ions, which can therefore cause mutations or break the DNA backbone and chromosomes. With non-ionizing radiation, this includes UV light. This will put in enough energy that it can actually cause adjacent thymine bases to covalently bond and make thymine dimers. This will prevent hydrogen bonding with the adenine and ends up distorting the sugar phosphate backbone and ends up breeding a different base pair. Chemical mutagens are going to be chemicals that will induce mutation, and there are several of these. These are just a few examples. With nucleotide analogs, these are going to be structurally similar to normal nucleotides. These can be incorporated into the DNA, and then they will inhibit polymerase and mismatch pairing. Some examples are 5-bromouracil and 5-FU or 5-fluorouracil. These are used as anti-cancer and antiviral drugs. Because of causing this interference and interruption in translation, these are very toxic drugs, and so they are only used in cases where it's absolutely necessary to be able to destroy the DNA, such as cancer or severe viral infections. These would not be used to fight bacterial infections where you have something else as an option. Other nucleotide altering chemicals, these can alter the structure of the nucleotide. So nitrous acid can remove the amine off of adenine and convert it to a guanine analog. So you would have an AT pair change to a GC pair. Aflatoxin, this is in a mold that will occur on grains. This converts guanine into thymine, so a GC pairing is converted to a TA base pair. Aflatoxin is also known to cause cancer. Frame shift mutations, these insert or delete nucleotide base pairs, or they can slip between adjoining nucleotides. So benzopyrene in smoke can cause this. Ethidium bromide, which is a stain used on DNA, can cause this, and acridin is a mutagen that's used in genetic research. So most of us are exposed to benzopyrene in smoke on occasion. With the ethidium bromide, these types of things used in the lab do have to be used in a very controlled setting where a person using these things needs to be extremely careful to avoid risk and exposure to themselves from using these types of chemicals in the lab. So the frequency of mutations, they're usually seen in 1 in a million genes. Mutagens will increase the rate 10 to 1,000 times and induce error in 1 in 1,000 to 1 in 100,000 genes. So fortunately, we do have some mechanisms to repair DNA. Thymine dimer repair in prokaryotes. The DNA photolyase is activated by visible light to help break the bond between the thymines and the thymine dimers. So light repair is activated by light, and it helps to reverse the mutation and restore the original sequence. Dark repair in the prokaryotes occurs in the dark or light. It's going to cut out the damaged section from the DNA, and it gets repaired by DNA polymerase and DNA ligase. With base excision repair, an enzyme will come along and cut out the erroneous base, and then the DNA polymerase enzyme will help to fill in the gap. With mismatch repair, this scans the new DNA looking for errors to remove and replace, and it distinguishes the new DNA because the old strands have become methylated. Sometimes we can't make the repairs. So with SOS repair, the damage is too extreme to repair. So a new DNA polymerase that's capable of copying sequences with mistakes is used. This will introduce mutations into the population, but it does allow the offspring to survive. So a mutant cell is a cell that does not successfully repair its mutations, and all of its descendants are also mutant cells. Wild-type cells are the ones that are normally found in nature, and they're assumed to be the ones without mutations. So when you do positive selection, you are selecting for a mutation by eliminating the wild-type phenotypes in your population. An auxotroph is an organism that has a different nutritional requirement than its wild-type phenotype. This can be used for negative selection or indirect selection, where you will select for the auxotrophic mutants. This can easily be done by just altering the nutritional things that are available for the population that you're growing on your media. So this example is how you would go about the selection of a tryptophan auxotroph. So you would inoculate your bacteria that have these potential mutants onto a media containing tryptophan. The colonies grow, and you're going to get a mix of auxotrophs and wild type. You use a sterile velvet pad, and it literally is a velvet pad, that you will stamp onto the plate, and you transfer the cells from the colony. You stamp the velvet onto replica plates that have tryptophan and a replica plate that is without tryptophan. You're able to identify the auxotrophs growing on the media with the tryptophan, but not the media without the tryptophan. So then you can inoculate a colony of auxotrophs onto the media with the tryptophan. The Ames test is used to screen for mutants. It assumes that any mutagen is also a potential carcinogen. It uses salmonella for this test with a point mutation. So it is a His-negative auxotroph. These mutants are mixed with liver extract and the suspected mutagen. The reason for using the liver extract is the liver enzymes are thought to stimulate conditions in which the substance would become a mutagen. So a lot of substances in the human body are going to be detoxified in the liver, and it's trying to mimic that situation. If it causes mutations, some of the mutants will revert back and grow on media without histidine. So a carcinogenic substance is a substance that causes mutations that end up resulting in cancer. So genetic recombination is the exchange of two DNA molecules that are composed of homologous sequences. Your homologous sequences are going to be the DNA segments composed of identical or nearly identical sequences. The recombinants are the DNA molecules that contain the new arrangements of the nucleotide sequences. So in order to create more genetic variety in eukaryotes, we will have crossing over. This is where the homologous chromosomes will recombine and trade some of the genes while forming gametes. With vertical gene transfer, you pass a copy of the genome from one generation to the next, from the parent to the offspring. With horizontal gene transfer, this occurs in less than 1% of populations, and it's in prokaryotes. They can acquire genes from another microbe in the same generation. So that would be more on the lines with swapping genes with a sibling. The donor cell will contribute part of its genome to the recipient, and the recipient cell may be a different species or genus. So when we look at bacterial transformation, with transformation, the recipient cell takes up DNA from the environment, such as dead organisms. Cells that have the ability to take up this DNA are said to be competent. Transduction is the transfer of DNA from one cell to another via replicating viruses. Transduction can occur in both prokaryotes and eukaryotes. So phages or bacteriophages are viruses that will infect bacteria. So with the transducing phage, the phage proteins are going to incorporate some of the bacterial DNA along with the phage DNA when assembling to make its new phage particles. The phage will attach to the host and it injects its genome. The phage enzymes are translated by the host ribosomes and it degrades the host cell DNA. So here the bacteria is injected in enzymes or instructions to make these enzymes that basically takes the host cell machinery hostage. The phage is going to direct the cell to synthesize phage DNA and proteins. Then it will normally assemble the phage DNA and phage proteins, but sometimes it may mistakenly incorporate some of the bacterial DNA along with it. Then the host cell will lyse and it will release daughter phages and the transformed phages. So this will allow some of the normal phage to be passed on, but also these transformed phages that have some bacterial DNA that will go on to infect other cells. The recipient host incorporates the donated DNA into the chromosome by recombination. So in generalized transduction, the phage is going to carry random DNA segments from the donor and the host cell to its new recipient. With specialized transduction, only certain host sequences are transferred along with the phage DNA. So another way that bacteria can swap some genes is bacterial conjugation. Here the donor remains alive. So the conjugation pili is also called a sex pili. It's a protein rod that extends out from the surface of the cell that's used to transfer genetic materials. It's basically like a poker that sticks out of the side to poke and insert genes into the other bacteria. So the F plasmid is going to contain the genes coding for making a sex pili. The sex pilus connects to the donor and recipient cell. The cells touch and stabilize. The donor replicates one strand of the F plasmid and transfers it to the recipient cell. The F negative cell synthesizes the complementary strand and then becomes an F positive cell. So once it has received this plasmid, it has become an F positive cell. So HFR, high frequency of recombination cells, the plasmid does not remain in the cytosol, but it ends up integrating into the chromosome. And these can also conjugate with F negative cells. The F plasmid integrates into the chromosome. The HFR and F negative cell join via a sex pilus. It replicates the F plasmid. The new strand is carried to the recipient along with a copy of the donor chromosome. The cells generally separate before the entire chromosome is transferred. The recombinant integrates part of the donor DNA into the recipient. So transposons, these are sometimes referred to as transposable elements. These are segments of DNA that are from 700 to 40,000 base pairs that can move from one location to another in the DNA. Transposition is the action of a transposon. It acts like a frame shift mutation because it will just do a frame shift insertion. This occurs in prokaryotes, eukaryotes, and viruses. Palindromes are sequences that read the same forwards or backwards, and these are usually going to be on both ends of transposons. So you can think of a palindrome being like the word mom, where it's spelled the same, M-O-M, forward. If you spell it backwards, it's spelled the same. A palindrome is going to be the same thing with the sequences. So the inverted repeat is going to be a sequence of nucleotides that are identical to the inverted sequence in the complementary strand. So if you read it in the 5' to 3' direction, it's GAA-TTC. The complementary strand is GAA-TTC. However, you can see everything is binding with its complementary base pair as well. Transposase is an enzyme that recognizes its own inverted repeat sequence at a target site, and it's going to cut the DNA at that location, and then it will insert the transposon or a copy into the DNA at that site. Using these palindromes and inverted repeats, we actually can purchase enzymes for use in a lab, and this is one of the steps in allowing genetic manipulation. Is you can cut at specific sites and insert chunks of DNA in where you cut the DNA. With complex transposons, these are going to contain one or more genes that are not connected with transposition, such as genes for antibiotic resistance. R factor is going to convey antibiotic resistance. So biotechnology is the use of microbes to make practical products. And generally these products are made for human purposes. We're not real big on making them to help non-human things as much. With recombinant DNA technology, this is the intentional modification of an organism by natural and artificial means. So goals of genetic manipulation: to eliminate undesired phenotypic traits, combine beneficial traits of two or more organisms, and to create organisms that synthesize products humans need. We already do quite a bit of that, actually. Most insulin is actually created by genetically modified bacteria. With recombinant DNA technology tools, mutagens are used to induce changes in the genome, and then you select for the cells with the characteristics that you consider to be beneficial. One good thing that has come out of the HIV virus is the use of the enzyme reverse transcriptase. This creates a flow of genetic information in the opposite direction of conventional transcriptase. So conventional transcriptase is going to go from DNA to RNA. Reverse transcriptase is going to allow you to use RNA to make DNA. So we refer to this as cDNA, and it uses an RNA template to transcribe your complementary DNA, or cDNA. This is easier to isolate the desired genes on than mRNA, because you can have more copies of it, and in eukaryotes the mRNA still has to be modified, and so the mRNA will actually have the introns removed, and it can be inserted into prokaryotes to make eukaryotic proteins such as insulin. We can make synthetic nucleic acids. We can actually produce DNA and RNA in a cell-free solution. This can be used to find the genetic code of things, to create genes for specific proteins, and to synthesize RNA and DNA probes that are used to locate specific sequences. With a probe, what you would do is have a nucleic acid that has a specific sequence, and you label it so that wherever it finds its complementary sequence, its location can be detected. One of the common ways to do this is to use fluorescence or to use radioactive materials. Restriction enzymes, these are going to cut DNA molecules at locations with specific and palindromic nucleotide sequences. When you cut at a specific location, then you can insert a gene into that specific location. Vectors are going to be nucleic acids such as viruses, transposons, and plasmids that we use to deliver a gene into the cell. Some useful properties of vectors: they're small enough to manipulate in the lab, so they're easy to insert genes into. They survive inside the cell. They can contain a recognizable genetic marker and ensure genetic expression by providing the required elements like promoters. So when using vectors, a restriction enzyme is going to cut the DNA and the vector into fragments with sticky ends. You can insert in the DNA you would like at this point. The enzyme ligase is going to anneal the fragments into a recombinant plasmid. Then the plasmid is inserted into the bacterial cell. The bacteria is grown on a media that will select for that cell with the plasmid. So PCR, or the polymerase chain reaction, is used to amplify DNA in vitro. It's a repetitive process that will separate and replicate the DNA strands. During the denaturation phase, the DNA is heated to 94 degrees Celsius, and this separates the two strands of target DNA. During priming, you're going to lower the temperature down to 65 degrees, Celsius and add in a mix containing excess DNA primers and complementary sequences near the end of the DNA. DNA polymerase is added, and an abundance of the 4 bases are added. This will allow the double-strand DNA to reform, provides a 3' hydroxyl group required for the synthesis. During the extension phase, the temperature is raised up to 72 degrees Celsius to increase the rate that the DNA polymerase replicates each strand. So this used to have to be done manually with moving your mixes into different water baths at different temperatures and using a timer. Now we have thermocyclers that are actually able to fully automate this process so it can be repeated over and over. After just 30 cycles, which can be done in a few hours, you can produce over 1 billion identical copies of the DNA. This is actually really useful for anything where you have to do genetic research because it gives you enough to work with. So there's an animation of the PCR process available on here as well. So for clone selection of the recombinant cells, you're going to use probes to bind specifically and exclusively to complementary nucleotide sequences, and you would use a radioactive or fluorescent label. This would allow you to be able to see which cells actually have these sequences you want. So sometimes we want to be able to separate DNA, and this is done in one of your virtual labs. You're going to do gel electrophoresis for this, and it separates the molecules based on the electric charge, the size, and the shape. DNA has a negative charge, so it's going to move to the positive electrode through an agarose gel. The southern blot, this begins with gel electrophoresis, and then it's going to transfer the DNA onto a less delicate nitrocellulose membrane. And then probes are used to localize the DNA sequences of interest and create a fingerprint. So the name Southern blot actually came from the name of the scientist who developed it. His name was Southern. The northern blot is a similar technique, but it's going to be used to detect specific RNA sequences. This one, however, was not named for a scientist. They just went with the northern name because of the southern name for the scientist before. So when it comes to inserting DNA into cells, we have a few options. Electroporation uses an electrocurrent to put a microscopic hole in the cell membrane. Protoplast fusion uses protoplasts, which are cells with their walls removed. The protoplasts encounter one another and the cytoplasms fuse to form a single cell with the genomes of both cells. There are injection techniques that use a gene gun that's powered by compressed air or a blank .22 caliber cartridge. Or you can use microinjection using a micropipette. So you may have seen video clips of using micropipettes with artificial insemination in fertilizing eggs outside the uterus. So some applications of the recombinant DNA technology. Genome mapping, this allows us to locate the genes on nucleic acids. The entire human genome was actually mapped in 2003. With restriction mapping, you can map the relative locations of genes and then compare the DNA fragments that result from the cleavages by several restriction enzymes. And this will be used to determine each fragment's relative location to another after the fragments are cleaved. And their sizes are determined using gel electrophoresis. And here's a little video showing some restriction enzyme use. Genomics, this is the sequence and analysis of the nucleotide bases of the genome that are used to cleave and map them on a gel. Now we use the cDNA, so it's much faster. So medical applications of DNA technology. Protein synthesis, you can insert synthetic genes for things like insulin and interferon. Vaccines. Subunit vaccines are made by inserting antigenic polypeptides to make the proteins that will stimulate immunity without actually having to inject the organism. Subunit vaccine is going to be safer because you're just going to give the polypeptides to stimulate the immune system. The organism is not actually introduced into the body at all. Hepatitis B is one example of this type of vaccine. It can be used for genetic screening. You can use the southern blot to screen for the presence of a gene causing a disease. DNA fingerprinting. This will allow us to identify individuals or organisms by their unique DNA sequence. This can be used in paternity cases and forensics. Gene therapy is one of those things that is still in research that we would really like to be able to see used clinically more. Here, missing or defective genes get to be replaced with normal copies of genes. With xenotransplants, this involves the transfer of animal cells or tissues or organs into human tissues. You could potentially be able to inject a gene into the animal and have them grow human tissues or organs that would not be injected. That one does sometimes raise some ethical issues based on people's feelings about using organs or using animals specifically to grow things for human use. There's agricultural applications of recombinant DNA technology. It's used to produce the transgenic organisms, recombinant plants and animals that are altered for a specific purpose. So to create herbicide resistance, one example is inserting a gene from Salmonella that conveys resistance to glyphosate or Roundup. Made organisms that have salt tolerance. Excessive salt in the soil can decrease the farmland, and if you insert a gene for salt tolerance, those organisms can use the salty soil, and the organism can actually end up removing the salt to help restore the soil to be used for other things. We've created some freeze resistance using a strain of Pseudomonas that has a polypeptide that prevents ice crystals from forming, and this can be sprayed on crops. With pest resistance, the Bt toxin from Bacillus thuringiensis is going to kill insects. It gets inserted in a variety of crops. We can use this to manipulate the nutritional value and yield. Bovine growth hormone has been used for cows. It's grown in bacteria, and it will cause additional milk production in the cows. We have ways of increasing shelf life of foods. There's a gene for beta-carotene that's been added to rice to try and increase the nutritional value. So recombinant DNA technology has lots of issues for a lot of people with ethics and safety. Some questions. These are not all the issues by a long shot, but just some ideas of some issues that have been raised. Is it ethical to raise animals solely to grow human tissue? The long-term effects are not really known. We haven't been doing this all that long. Gene transfer may end up in creating some organisms that we don't want to have this gene growing in. We may create some new pathogens. The technology is used for bioweapons, so it does get in the hands of people who make weapons with it, and we can't really prevent that from happening if we are teaching people how to do this type of thing. We may create new proteins that we don't really know how to deal with. Looking at genetic screening, how should we handle it? Is it going to be routine or not? Who would pay for it? Who knows the results of it? It takes issues with HIPAA much further. Should we be able to choose the sex of a child? Right now we have the capability of doing it, but generally it is not considered an acceptable practice to choose the sex of your child.